So I'm primarily going to talk about some data that we published a few weeks ago in Scientific Reports, where we reported that we may be able to restore normal embryogenesis in eggs which were deficient of mitochondrial DNA by using a mitochondrial supplementation process. And this work has been primarily done in a pig model that we've been developing over the last few years. So by way of competing interest, I need to declare that the work that I'm going to present to you today was primarily funded by Oversicience to a research grant that was awarded to my laboratory at the Hudson Institute of Medical Research. So what is the mitochondrial genome? Well, it's between 16.2 and 16.7 kb in size across the large mammalian species, and it encodes 13 of the proteins of the electron transfer chain. And interspersed between each of these coding genes, there are tRNAs, in total 22. There are also 2 rRNAs, and there's one non-coding region, which is the D-loop region. And this is the site of interaction for the nuclear transcription and replication factors that translocate to the mitochondrion to drive first transcription and then replication of this bacterial originating genome. The D-loop also has 2 hypervariable regions which are used by geneticists to map the maternal-only transmission and inheritance of this genome. Now, mitochondrial DNA copy number is very strictly regulated during development. The primordial germ cells, which are the very first germ cells that are laid just after gastrulation, have approximately 200 copies of mitochondrial DNA each. And during the process of oocytes, as these cells differentiate and into mature oocytes, mitochondrial DNA copy number increases exponentially from this low level of template that is available. And in fact, it's becoming clearer now that the mature egg, which has fertilization capacity, has approximately 200 copies of mitochondrial DNA. Following fertilization, there are significant decreases in mitochondrial DNA copy number up to the blastocyst stage. And it's becoming clearer now that this decrease and even culling and excretion of mitochondrial DNA into the extracellular environment is a good indicator of good embryonic development. But at the blastocyst stage, there is a major replication event, and this is specific to the trophectoderm. So all naive cells will want to have as few copies of mitochondrial DNA as possible. This enables them to proliferate very quickly, divide very quickly, to produce copies of themselves. And this is done under the influence of glycolysis. OXPHOS slows down that process because it's much more of a longer-lasting process to generate those 33 to 34 molecules of ATP that are derived from each of the 2 molecules of glucose that are invested. So what happens is that the inner cell mass continues to divide out its mitochondrial copies. And these are the cells which give rise to the embryo proper and to the fetus. And what then happens is that something known as the mitochondrial DNA set point is established. And this is where naive cells have very, very low levels of mitochondrial DNA copy. And they maintain this low level of mitochondrial DNA copy until they commit to specific lineages. So this then enables heart cells and muscle cells to have high levels of mitochondrial DNA copy because these cells primarily use oxidative phosphorylation and the genes from the mitochondrial genome which are encoding for the electron transfer chain, while blood cells and sperm cells, for example, primarily use glycolysis, and they would therefore not have a great requirement for OXPHOS and therefore do not need large numbers of mitochondrial DNA copy. But establishing the mitochondrial DNA set point also then allows the primordial germ cells of the next generation to achieve that target of 200 copies, which are then cycled in a maternal-only manner from one generation to the next through these early germ cells. And that's primarily how we inherit our mitochondrial DNA. So we, like others, have been highly interested over the last few years to determine whether there was a relationship between mitochondrial DNA copy number and fertilization outcome. And in work that we published way back in 2006, we, like others, have shown that those eggs which have the potential to fertilize, have significantly higher copies of mitochondrial DNA than those eggs which remain unfertilized. And this is irrespective of whether those eggs that fertilize give rise to 3 pronuclei or 2 pronuclei. The difference is significant compared to those that arrest at the 2PN stage. So, back in 1997, Jacques Cohen recognized that there was a problem for many women, older women, that their eggs quite often arrested during early embryonic development. And he proposed supplementing these eggs with an exogenous source of material, cytoplasmic material, that came from a young donor egg. And he introduced this material into the egg with the hope that he would be able to firstly rescue embryonic development and secondly produce live offspring. And indeed he did this, but this resulted in heteroplasmic offspring. In other words, offspring that had inherited 2 populations of mitochondrial DNA. So in essence, these offspring had 3 parents, chromosomal mum, chromosomal dad, and 2 mitochondrial mums. And then it was subsequently reported that there were likely to be some developmental disorders resulting from this approach. And governments around the world and their the regulatory authorities started to impose bans on the use of this technology. Back again in 2006, we were interested in whether it would be possible to supplement mitochondrial DNA deficient eggs, in other words, eggs with low mitochondrial DNA copy number, with mitochondria originating from eggs, from sister oocytes. In other words, eggs that had been harvested from the same ovary. And indeed we did this, and we were able to increase fertilization outcomes as a result. The model we chose to do this on was our BCB model in the porcine system. BCB stands for brilliant crescent blue, and it's a basic dye that enables you to label eggs when you harvest them from an ovary prior to in vitro maturation. Those eggs will either stain positively, and we call them BCB positive, or BCB negative are the ones that stain negatively. And effectively, BCB is informing you about the state of developmental competence of those eggs. So those eggs which are BCB positive are developmentally competent. They cease to express the enzyme glucose 6-phosphate dehydrogenase, and therefore are unable to break down the label brilliant crescent blue. While those eggs which are developmentally incompetent are still expressing the enzyme and are able to break down the label. And consequently, we have 2 populations of eggs. Not only can we distinguish the eggs based on the staining by BCB, but there is a difference in terms of these eggs, in terms of their cytoplasmic volume, with the cytoplasmic volume being larger in these than for those. When we look at mitochondrial DNA copy number in these eggs, we can see that the BCB positive eggs have high levels of mitochondrial DNA copy number when we collect. During the final stages of maturation, there's a culling and then an increase in mitochondrial DNA, so that the levels are restored by the metaphase 2 stage. But when you look at the BCB negative eggs, you see the inverse profile, lower copy number at collection, higher at 22 hours of in vitro culture, and then very low levels of mitochondrial DNA copy at the metaphase 2. And that's for those BCB negative eggs which manage to get to the metaphase 2 stage. And indeed, when you compare copy number in these 2 populations, you can see that for the BCB positive eggs, mitochondrial DNA copy number is significantly higher for the BCB positive compared to the BCB negative populations. So we've effectively used this as our model. But just to emphasize the importance of the model further, we've used an agent which depletes cells of their mitochondrial DNA during in vitro culture. And you can see that when you culture BCB positive eggs with this depletion agent, firstly, after 22 hours and then at 44 hours of culture, you're losing mitochondrial DNA integrity. And the reason for that's happening is that you've got a steady reduction in mitochondrial DNA copy during the final stages. So when we get to the 22 hours where we see a decrease, we get more of a decrease. But what's interesting is that because the agent's there, we cannot reinstate mitochondrial DNA copy. And then when we fertilized those, attempted to fertilize those eggs which had been treated with the depletion agent, they either failed to fertilize or the embryos arrested during pre-implantation development, which sort of provides experimental evidence that mitochondrial DNA copy number is important in this process. So what we really therefore have is a model where we have BCB positive eggs which have the mitochondrial DNA copy number is above the threshold that is there for fertilization outcome. In our model, that's around 150,000. And we have a population of BCB negative eggs. Those eggs which are BCB positive have sufficient mitochondrial DNA investment to support these subsequent developmental events. But the BCB negative eggs do not have sufficient mitochondrial DNA and are not able to support subsequent developmental outcomes. So the question we've then gone on to ask is, could we, using this model, could we supplement these eggs with a small amount of mitochondria that we can get into an injection pipette and mediate this event here? So in other words, create something to happen with this small investment to support this larger scale investment later on during development. And indeed, that's what we did. We took our BCB negative eggs and we isolated mitochondria from our BCB positive population, and we injected this population of mitochondria into the egg, a technique which we've now called mitochondrial transfer during ICSI. And in fact, the amount of mitochondrial DNA that we're investing is around 800 copies. So what happens to that mitochondrial population as we inject it in? So here you can see some confocal microscopy where we've labeled the mitochondria with mitotracker green, with TMRM and mitotracker deep, and the egg itself with mitotracker deep red. And as you can see here, this is the bolus that was injected in. It's present at 1 hour, and you really have to blow it up to see that the mitochondria is really present there. And again, that mitochondria is persisting at 24 hours after injection. And it's still in that bolus shape there. So it hasn't started to disperse at this stage across the cytoplasm, and it doesn't appear to be a mitochondrial replication event that is actually taking place. So here are the developmental outcomes. When we take BCB negative eggs and we fertilize them using normal IVF means, we get, with a BCB negative population, we get around 7.6% fertilization outcome. However, when we perform MICSI, we get a blastocyst rate of around 31.5%. And this compares with the outcomes that we see with BCB positive eggs that were either injected by ICSI or MICSI. They have blastocyst rates of around 33% and 31.5% respectively. So what we're seeing is similar rates for our mitochondrial DNA deficient eggs at the blastocyst rate as we are for the positive ones when they're supplemented. And in fact, supplementing BCB positive eggs with extra populations of mitochondria is not helping developmental outcome as far as we see at the moment. We looked at numbers of cells present in our MICSI-derived BCB negative blastocysts, and what we saw was that there was an enhanced number of cells present at the blastocyst stage when we compared the outcomes with those BCB positive blastocysts generated through ICSI and the BCB negative blastocysts derived through ICSI. We then had a look at mitochondrial DNA replication profiles during pre-implantation development, and we were able to endorse our previous finding that we saw in the BCB positive populations a decrease in mitochondrial DNA copy number during pre-implantation and an increase in copy number at the blastocyst stage. So that was data we originally published in 2007, and we've further endorsed its validity. what we see for the BCB positive population of embryos that were generated through ICSI is that we get a decrease in mitochondrial DNA copy number early on, but the profiles later on during pre-implantation development are very different, which goes some way to suggest that there are some developmental delays associated with ICSI, and perhaps this supports that hypothesis. When we take the BCB negative embryos that were generated through ICSI, we see that mitochondrial DNA replication events are really playing very much a catch-up process here. That copy number is slowly increasing towards the blastocyst stage. But that's in those eggs which successfully develop, and that's not always the case. However, when we look at the BCB negative population of embryos that were generated through MIXI, we see this significant increase in mitochondrial copy number between fertilization and the 2-cell stage. And this ensures that there's enough mitochondrial DNA present at the blastocyst stage. And if we look at all 4 treatments together on this graph, you can see that if you compare the copy number that's present in the metaphase 2 oocyte for the BCB negative eggs and then at the 2-cell stage, you can see there's around a 4.5-fold increase in mitochondrial DNA copy number. But all 4 treatments seem to have the same levels of mitochondrial DNA present at the blastocyst stage. But what was interesting for us was that this increase in mitochondrial DNA copy number was happening prior to embryonic genome activation. So the question we were interested in asking was, were we actually affecting the health and viability of the embryos that we were producing by initiating a process that happened prior to embryonic genome activation? So what we did was that we performed single blastocyst microarray assays, whole genome microarray assays, where we looked at blastocysts that were generated from 3 different approaches. Firstly, we generated blastocysts from BCB positive eggs that had been produced by ICSI. We then took BCB negative blastocysts derived from BCB negative eggs that were generated through ICSI, and also the BCB negative derived blastocysts that were generated through MIXI. And what we saw was that there were very different gene expression profiles as a result. But those blastocysts which had been generated through MIXI and were from BCB negative eggs, their gene expression profiles were much more similar to those blastocysts which had been generated by ICSI from BCB positive eggs, while the disparity in gene expression was much more on the side of those blastocysts from the BCB negative population that had been generated through ICSI. And when we looked at the specific genes that were affected, we saw upregulation of many genes from the BCB negative derived blastocysts from ICSI associated with metabolic disorders such as diabetes, obesity, and amino acid regulation, and also mitochondrial metabolism, suggesting that these blastocysts, which are lacking mitochondrial DNA prior to the embryonic genome activation stage, perhaps are associated with severe metabolic disadvantage. So, just to conclude, we're beginning to think that mitochondrial DNA is important for developmental outcome, and having sufficient present in the egg at the time of fertilization can lead to better blastocyst development and hopefully a better chance of a take-home baby. But if there's insufficient mitochondrial DNA present, fertilization events or embryonic arrest are hindered, and consequently blastocyst development is unlikely. In the case where blastocysts are formed, then it appears that these are disadvantaged by factors related to diabetes and other metabolic disorders, and that has consequences for the health of the offspring in the long term. But if you take these BCB negative eggs, or those which are deficient of mitochondrial DNA, and you supplement them with an autologous population of mitochondrial DNA, you initiate a mitochondrial DNA replication event which significantly increases mitochondrial DNA copy number. You appear to be resetting the embryonic genome, which should produce healthier and more viable blastocysts, and hopefully with a greater chance of a take-home baby. And I'm sure Bob Casper will go into a lot more detail about this later on during his talk. So I'd just like to finish by acknowledging co-workers who've been involved in this work. So Gail Kagoni, Tisha Sai, Yogesh Makajani, Pam Matthews and Matt McKenzie, who are based with me at the Center for Genetic Diseases. Shahi and Emma, who did much of the early work back in 2006 and provide the foundations for what we're doing now. And to our collaborators at Harvard, David Sinclair and his co-worker Michael, and David's group also at University of New South Wales in Sydney, Ashley and Lindsay, and to our platform technology helpers Kirsten and Jody at Monash University.